In this short video, I'm going to discuss the one-ring, two-target protocol, which is part of the cell biology protocol collection. To start, select the count size ribbon and press the protocols button. Select the cell biology group and scroll down to the ring, one ring, two targets protocol, and press the load button. The protocol is designed to create a ring mask around the nucleus and then measure two individual targets within that mask. Next, we're going to load an image. All protocols come with demonstration images, which next to the reset button may be loaded through the dropdown by selecting Load Demo and having the Open Demo image checked. You may use your own images as required for your own experiments. Next, we're going to set up the experiment. In this case, we're going to select the host channel for the parent. This can be edited, and you can put in nuclei if you wish, or any other nomenclature. And then select the two targets. In this case, we're going to use the GFP target and use the size 7. With the interactive selection checked, press the next step button. Now we're going to segment the nuclei. In this case, it's set up for using the histogram-based segmentation. However, we can use smart segmentation also. I'm just going to simply select the OK button, and then we can adjust our parameters. In this case, the ring is set to 15 pixels. As you can see, we've grown out 15 pixels from the nuclear area. We can adjust that by simply changing this value. I'll bring it down to 10. And press the apply button. As I make changes, you always need to press the apply button when it's active as you make a change. We can also get the perinuclear region by adjusting the inner line. In this case, I'm going to set it to 4, -4, and press apply. So now I've brought in the ring mask 4 pixels into the nuclear area, and it's 10 out from the nucleus. So we now have a 14 pixel ring. You have other options. In this case, the minimum area was if you have a broken up nuclei, other debris, you can segment that out. We can run clean borders, fill holes, and we have a few splitting options for none, watershed, and boundary shape. Adjust these as needed for your particular data set. Next, we'll press the next step button. And now we'll start segmenting our targets. In this case, you can see where we've created the perinuclear ring around here. You can simply, again, use threshold segmentation, adjust accordingly to your own analysis needs, or use smart segmentation. I'm going to press OK. Again, we have options to filter out or gate out any small areas, fill holes, that's off, or object splitting. Press the next step button. Again, now I will segment my second target. It's actually pretty good as it is. I can turn the show off and on just to show how well it's segmented. You'll need to leave this button on and show the overlay. Press the OK button. I have no options to adjust for this step. And now we'll press the next step button. All the values are now calculated on a per cell basis. You can see the results. We have additional data tables available to show the statistical information. As part of another video, we go through a full explanation of how to adjust the data tables, how to show the data, and whether to automatically export it or not. And as always, if you make changes for your own specific image sets, you can press the Save As to make that a permanent change, and that will show up as another protocol under the parent group, in this case, one ring, two targets. Next, we're going to press the Finish button. So we've now set up, tested, and validated our settings. At this point, we could go through and test a few more wells in this case, or images if they were in a folder, or we could just run a batch analysis by simply selecting what rows and what columns or what region of wells you'd like to analyze, take the protocol out of the interactive mode, and run selected. In this case, I'll just simply press the button. It will go through, it will reset the data tables and analyze both wells automatically, and then press finish. And now I have the data for both wells. At this point, you can now export the data.